Okay, so we are going to continue on the lecture. I think this is on 16, and we are going to talk about a little bit of the technique, functional knockout, and anti-sense. And I think and then we'll continue on the you know uh, signal 1, 2, 3, 4. I think uh, and then we'll just finish up the mesotherm induction and the formation. I mean, the patterning and everything. So this is about the antisense oligo. You inject antisense. Another name is, I think that's RNAi these days. I mean, RNAi actually came from this. So basically, you have an antisense RNA. Let me go to the previous next one. See, this is the sense RNA, mRNA sequence. Then antisense is that you have the complementary sequence. Okay, right. And then I think that's the you have this is the complementary sequence that is still sense. So I think that you change that. You will make this into the you reverse it. Okay, so that is the you know a 5' end is u. So look at this. This come from here, and then you just reverse it. Okay, so complement and reverse it, and that becomes the antisense. So what will happen is that you make this oligo, you synthesize this oligo, and then you put it in a cell. Then what will happen is this will bind to here because 5' is right here. So it will go to here, and then it will go to here, and then it will go to here. So this guy will bind to the sense RNA. So what will happen is that if if that happens, so antisense RNA binds to mRNA, then that mRNA is dead. So double-strand RNA is not tolerated. So cell will just degrade off that the double-stranded RNA. Okay, so we talked about it. Okay, so what you can do is that, okay, so in XNR is the passing all these 4 criteria. The last one was the functional blocking. So in this experiment, we have seen before, isn't it? The try to find out number 1 and number 2 signal. And if you inject antisense XNR 1, 2, 4, then what is it? What happens that mesotherm induction did not happen. And I think the same thing will happen, maybe BEG-T, you can do that earlier, then endotherm doesn't form, so it's not going to happen also. Okay, so, they try to find, XNR activity further, okay? So they did a XNR protein, and then did a chemical assay. So of course, it will turning into a mesotherm. So these are the Goosecoid, CHED, X-Wnt8, and XBRA. These are the probably mesotherm or like also dorsal marker. And what will happen is that, okay, you increase the dose of XNR, and then they will make this XBRA right here. This is true mesotherm marker. And then as you increase the XNR, and then here, and then it will produce more and more of the XBRA. And then Goosecoid, Cordin, these are the dorsal marker, okay? So they are, expressed. I think this is not very sensitive, but I mean, a high dose, you are able to, induce this, right? And then X-Wnt8 is the also mesotherm marker. Only this is the just like a ventral mesotherm marker or something. And then I think that they are sensitive to certain concentration, but they are still able to form, sensitive XNR concentration. Okay, so looks like what it is that let me go back. So what does it tell you is that this is the dorsal mesoderm marker. Okay, mesoderm marker. So what it means is, okay, so XNR is really inducing mesoderm, but the what it is that if you increase the amount of XNR, and then what you see is that you will see more of the dorsal mesoderm. And then X28 is a general mesoderm patterning, ventral, it's kind of like a ventral mesoderm marker. And then if it is too high, then the let me use the different colors. If the XNR is too high, then they don't produce X28 at all. So probably the ventral marker is lost. So they did, this is like such a tedious microsurgery you can ever think about. Okay, and that is they try to measure this gradient of XNR. So here's what they show. So you have the in this gel, okay, you made the you extract XNR this way. So from this, so you have the embryo, and then you cut it in the middle. Okay, oops. Okay, you cut it in the middle, and then you have a ventral hemisphere and then dorsal hemisphere. You grind it up, and then you load it. Okay, and then you look for the XNR. So XNR 1, 2, 3, 4, and then this is VEG1 is the control, and then this is the loading control. They saw it, and which part has the more XNR, isn't it? You will see is dorsal more. Dorsal has more extra, XNR. And then what they did is that they cut it in this way. Okay, so endoderm, marginal, which is a mesoderm, and then animal. In animal, XNR doesn't produce, okay? And vegetal, it is produced a lot. Of course, it's coming from mesoderm, right? Endoderm, right? And marginal, also they are producing. I mean, like when I talked about, what was it? The community effect, right? And then, mesodermal cell also produces the induced. Okay. And what they did is that, okay, so since the, vegetal here produces a lot, so they did a microsurgery on this vegetal chunk, and then those vegetal chunk also they divided into half. So ventral vegetal, and then dorsal vegetal. Wow, such a tedious, experiment. And what they found is that, okay, when they loaded, and then the dorsal vegetal produces much, much more than the ventral vegetal. Okay, so which means is that, okay, let's take a guess. So we, in previous slides, somewhere before, okay, and we, discussed that, okay, there's a number 1 signal and number 2 signal. And number 2 signal is coming from dorsal endoderm, dorsal vegetal, isn't it? And they're coming from Newcomb center. This is the Newcomb center, N center. Okay, right. So Newcomb center is producing XNR, and then more than the on the left, ventral vegetal. So that is kind of a smoking. So from that, maybe number 1 signal is same as number 2 signal, isn't it? But the then how the number 1 and number 2 signal? So number 1 signal is ventral mesoderm, right? And then the number 2 is the dorsal mesoderm, right? Okay, induction, right? But the then it is maybe the same signal, same chemical is doing that. And then the how they different is that number 1, number 2 is just more. So same chemical, but for number 1 and number 2, just number 2 gets more, and then they become dorsal, and then become ventral. Okay, I think that's where getting it. Okay, so you know, that is the true. You know, so maybe really what they found is a nodal related protein, and then nucleus center they just produce more. So that's why it become dorsal, and then it become ventral. So how they do that? What do you mean by more? If it's more, then what is more? And then how the more signal is different from less signal? I don't know, man. Okay, it's not really explained very well. I think that we just need to accept as a fact. Okay, I'm sorry. Okay, and I'm gonna move on. Okay, so after mesoderm is induced, okay, and then what will happen is that all mesoderm will express the transcription factor, that is extrabrachyury. Okay, and then the on the dorsal side, on top of this brachyury, and then they'll produce glucoid, X0, and X-LIM1. These are all transcription factors. Okay, so you know, this is the mesoderm, the slice of the you know, embryo, and then you can see mesoderm is forming like this. I wonder what is that? Probably you may, you know, you may be surprised, but this is the slice. So the real mesoderm looks like this, you know, something like this. Okay, it's kind of like a donut. So you know, donut ring kind of structures. Okay, right. So, but this is just only one slice. That's why it's showing here. So this is the tissue that produces expra. So that is the mesoderm. Okay. Okay. So you know, we took care of number 1 and number 2. They are all X and are 1, 2, 3, 4 or something. But the number 2 and number 1 signal difference is that just the amount, just the you know, signal, you know, the amount. So number 2 is more and number 1 is little. Okay. Then we need to deal with this, you know, number 3 and number 4 signal. Okay. So what they are saying is that they are crashing each other. So, you know, what do you mean by crashing it? So, you know, they did, you know, they tried to find the difference between number 3 and number 4. So they did, you know, very like a veggie one kind of approach. So they took the this dorsal lip, which is a Spearman organizer. Okay, I mean there are so many different things. Dorsal lip, we're gonna talk about it in like a lecture 19 or lecture 20 or something. So from dorsal lip, they isolate, grind it off, and then isolate mRNA, and then copy to a cDNA, and then make a library. And then there are so many different genes that's specific to a dorsal lip, okay? And then 1, 2, 3, 4, 5, 6, 7, 8, 9, you know, so many. And then you just inject into the you know, opposite side of the embryo, and then you know, keep on you know, checking whether they are able to create this kind of bump, so that you know, somehow it's a dorsalizing activity or something like that's coming from. So that's how, you know, they found this organizer molecule, and this many, ooh, a lot, right? And they have the ability to, you know, uh, able to, you know, create one more axis on the other side. Okay. This is the secreted protein that we are going to focus, okay? So I will stop here, okay? And then I'm going to continue on next segment. Thank you.